Boa tarde a todos. Hoje nós teremos uma tarde muito especial, não com um, mas com dois convidados, diretamente da França, tratando de um tema extremamente intrigante, eu acho que para todos os biólogos, né, que é esse ambiente aí estranho, né, de hidrogênio, desde a sua geologia até os micro-organismos capazes de sobreviver. O seminário será em inglês, nossos convidados são franceses, mas será em inglês, so I will speak a little bit in English just to thank you, both of you, to stay with us, bringing us this intriguing theme. And now I pass the speech to Dr. Krieger that will present our guests. Thank you very much.
Thank you. So it's my pleasure to introduce, first of all, Fabrice Brunet. He's a researcher at Grenoble Alpes University. Grenoble University is a part of the Institute of Earth Sciences. So his research focuses primarily on the physical and chemical studies of the planet Earth. Fabrice is a long-time collaborator, not only with my group at the University of Brasilia, but also a collaborator for our internationalization program, CAPSTREAM. So he was the first one that will talk. And we followed by Dr. Marianne Quemeneur. She's a researcher at the Méditerranéen Institute of Oceanography at Marseille, France. Marianne is a team member of the Environmental Microbiology and Biotechnology group. And of course, this is her main research focus. So it's my pleasure to have you here today. And you, without further long, pass the word to Fabrice. I think is the first one to talk. And today's talk will be the title will be Natural Hydrogen Environments from Geology to Microbiology. Thank you again for your presence.
Thank you, Ricardo, for the introduction, and thank you to the organizers to give us the opportunity to give this presentation. So basically, the subject of this presentation that we are going to give together with Marianne is an example of strong interaction between inorganic processes and living organisms. And I will talk about the geology, the inorganic processes, and Marianne will talk about the microbiology, since she is a microbiologist and I'm just a geologist. So the process that we are going to describe deals with the production of hydrogen in a natural system, in natural environments. And so according to inorganic processes, and Marianne will tell you how microorganisms they develop in such a hydrogen-rich environment. So basically, why study hydrogen? The first question we can ask to ourselves is, hydrogen really a green energy? Because you may have heard that, I mean, this is a promising fuel for energy, and it is true in one sense, because as you can see on this slide, this reaction of combustion of hydrogen with oxygen produces water instead of CO2 in the case of fossil fuels. And the energy that is delivered by this reaction is pretty high. And so basically we can compare hydrogen energy to gasoline, for example. So the specific energy of hydrogen is about 3 times the specific energy of gasoline. So it's really promising. Hydrogen can be used in different ways. It can be used directly in combustion engines, as you can see here. So just by this combustion reaction, and then you replace the gasoline in the same type of engine. But you may have heard about fuel cells, which is the same kind of way to recover the energy of this reaction, but it's not a direct reaction. It's a reaction through an electrical cell, which is a fuel cell using electrochemical energy. And basically, the energy that you get is electricity, and you can run a car with this kind of electricity. So now, what is quite interesting with hydrogen is when you look at the way hydrogen, industrial hydrogen is produced. And if you look the way it is produced, then you understand that it's not really a green energy, because 96% of the hydrogen that we use nowadays is produced by fossil fuel reforming. So, I mean, fossil fuel reforming, I can show you the simplified reaction of fossil fuel reforming, which is this reaction where methane reacts at high temperature with water to produce hydrogen plus CO2. So you see that this fossil fuel reforming production of hydrogen produces a lot of CO2. So it's not really a green energy in that sense. So you see when you produce 8 grams of hydrogen, you produce 44 grams of CO2. And only 4% of hydrogen is produced by electrolysis, so using electricity. And this electricity might also come from fossil fuel. So it cannot be considered as a fully green energy. So there is a way to produce a kind of green hydrogen, is to use renewable energies like windmills, or you can use the sun in order to produce electricity, to run an electrolyzer, and the electrolyzer can electrolyze water, decompose the water molecule, produce hydrogen, and this hydrogen can then be used later on into a fuel cell, what I presented in the previous slide, in order to produce electricity when you need it. Because as you know, the windmill works when there's wind. So the idea is to store the hydrogen and use this hydrogen when you need it. The problem is that I told you that hydrogen has a high specific energy, so a lot of energy per kilogram, but it has a low volumetric energy because it's a gas, so it takes a large volume, and there's not so much energy per cubic meter. And then the problem with this type of system is you have to store the hydrogen, and storing a gas, it's a real issue, and there's still some research work to be done on hydrogen storage, and people are now thinking of storing hydrogen underground in, for example, in salt cavities or in deep aquifers. So, but this is not really working so far. So what I want to show you is that it might be that there are some natural hydrogen sources that are available for industrial applications. And I will first show you the way natural systems are able to produce hydrogen. So there's one way that you all know, and Marianne will talk about it in a few minutes, which is the microbiology. I mean, as you know, there are some H2 producing enzymes like hydrogenase, like nitrogenase. So this is a way to produce hydrogen. And some people try to use this way to produce hydrogen for the industry, but so far it's just a small production. It cannot produce the amount of hydrogen that we need for the industrial applications. There's another way hydrogen is produced in nature. This is through the natural radioactivity. So this is something that happens, as you may know, natural rocks, they contain radioactive elements like uranium, like thorium, like potassium. And when these elements, they decay, they produce particles like alpha or beta or gamma particles. And these particles, they can interact with water. When you have, for example, rocks in contact with water in a deep aquifer, for example, then these particles, they react with water. They form what people call radical species. And this is a way to decompose the water molecule and to produce hydrogen at the end. So this is the way hydrogen is produced in nature. It's not a big flux of hydrogen that is produced this way, but it's a way hydrogen is produced. And the way I'm mostly interested in is another way. This is through geochemistry. So basically what we have quite recently discovered, like let's say 20, 25 years ago, is that, I mean, you're not geologists, so I will detail a little this map. So you recognize your country somewhere here. And what you see here is the mid-Atlantic oceanic ridge. And in this place, you have some high tectonics. So you have seismicity, you have some volcanoes on that place. And this ridge, which is a kind of a mountain on the sea, along this ridge, people have analyzed some fluids that are hot or cold fluids, depending. Marianne will probably detail this later on. These fluids have been analyzed, and what people discovered is that these fluids contain dissolved gases, hydrogen, which compose about 40% of the gas fraction, methane, about 6%, and another list, another few gases that compose those fluids. So it means that along this North Atlantic mid-oceanic ridge, you have gas that is venting and that contains quite a high amount of hydrogen. So people have been trying to find out how this hydrogen is produced, especially in a fluid where the temperature is so high that it could not be organic and could not be related to living organisms. So it has to be some kind of inorganic process. And a lot of work has been done, especially in the laboratory. People have done a lot of experiments to understand this. And what turns out is that this hydrogen is produced through abiotic redox reaction. And I want to make it very simple, but basically what happens is you have, in this part of the ocean, you have specific rocks, and they contain iron, which is 2+. These rocks, they react with seawater at high temperature, and then the iron is oxidized. And if you oxidize the iron, you reduce the water and you produce hydrogen. And this type of reaction is called serpentinization, because this type of reaction leads to the production of a rock that is called serpentinite. And basically, this is the way hydrogen is produced. And then, I mean, it is even more complicated, all these chemical reactions, because if you produce hydrogen, since you have CO2 dissolved in the seawater, you can start to react H2 and CO2, and maybe you can produce some organic molecules, so some hydrocarbons like methane, that would be abiotic methane. And all these molecules, as you know, I mean, hydrogen, methane, they could be a fuel for life. And some scientists think that the origin of life is basically in this type of environment where H2 and CH4 can be produced abiotically. So this is very interesting on the scientific point of view. On the industrial point of view, it is not so interesting, because these vents, these what people call the smokers, this is located at more than 2,000 meters under the sea level. When you are a geologist, you know that this kind of rocks that you find in the middle of the oceans, you can find them also in the middle of continents. I don't want to take too much time on this. This is really geology, but just to tell you a few words, because Marianne is going to talk about this type of environment. So here is a sketch of the crust. You have the continental crust, and here you have the oceanic crust. So this is kind of sectioned through this map. So here is in green, you can see the oceanic crust. We are in the case of what we call extension, because here is the case of extension, where the continent, they are pulled apart, and the ocean crust is growing. So it's an extension context. So here is the extension context, but sometimes the plate tectonics works in the other side. And if instead of having extension, you can have compression. And then what happens is that the oceanic crust tends to disappear. And sometimes it is squeezed within mountain belts. That's the very end of the convergence. That's the collision where you form orogenic belts, mountains, and you can have some oceanic rocks that are like squeezed within slices of continental crust. And if you, like here in Grenoble, we have the Alps mountains, and if you go hiking in the Alps, you may find these some examples of oceanic crust that is now at 2,000 meter elevation. And in this type of rocks, potentially they can still produce hydrogen according to similar reaction that the reaction that I introduced here. But now if the reaction occurs, it will occur at lower temperature because now the water is not the seawater, it's continental water, it's the meteoric water that interacts with this oceanic crust and can produce hydrogen. So there are some examples of the world of such slices of oceanic crust that is onshore. And here we probably don't see it very much, but these slices of oceanic crust, they are called ophiolite, and there are quite a lot of examples of ophiolites worldwide, where you have some springs with water and with gases. And here you can see the flame. This is in Turkey, and this is the Chimera sea page, where you have methane, and you see there's a flame that is related to methane, but you also have H2 in such environment. So you can have springs with H2 that is produced on the continents. So that's becoming interesting if you want to collect this hydrogen for industrial applications. Here, just an example of the good point when you have the arsenic crust, which is visible in the mountain belt, is you can see you have a wall section across the rocks, and this is the rock type that I described before, which is the serpentine, and you can see here a serpentine rock that probably produced hydrogen before. So this was related to oceanic crust, like on the sea or in the continent, related to ophiolite. But there are other settings that have been discovered quite recently, where hydrogen is produced as well. And I will just shortly give two examples. One is an example in Minas Gerais in Brazil. And this is, you can see this feature here, which looks like a Mickey Mouse a little bit. You know, you have the face with the two ears here. And in this location, some of my colleagues, they installed some hydrogen sensors, and they monitored hydrogen concentration in soil over time at about 80 centimeters depth. And this is what they found. You can see these curves. This is the hydrogen concentration as a function of time. And you can see that you have some pulses of hydrogen through the day. The maximum is close to, the maximum concentration is recorded close to noon. And you see it's quite a low concentration compared to what I've presented before. It's about a few or 100 ppm or a few hundred ppm of hydrogen, what is quite low. But in some places, some sensors, they recorded much higher concentration, up to 1,000 ppm. So, okay, this is quite small, but if you integrate the hydrogen flux in such a structure here, it's about 500 kilograms H2 per day. So it's something. And now people, we don't really know what is the origin of this H2. Is it biotic? Is it abiotic? Is there a deep reservoir of H2, a deep H2 accumulation where people, I mean, if we drill, do we get a pocket of gas that could be basically used? We don't know yet. What we know is that there's just one example in the world where there's a real accumulation of H2. And this example is in Africa, in Mali. It's about close to the capital city of Bamako, where it's an interesting history, because people in 1987, they drilled a well, and they had a big explosion, but they didn't know, I mean, what happened. They had a big explosion, so they plugged the well, and this well was unplugged like 20 years later. And they discovered that the reason for the explosion was a high concentration of hydrogen. And they discovered, quite big accumulation of hydrogen, because the gas that was analyzed contained 98% of hydrogen. And that's the only example so far of a deep reservoir. And actually, I think there are 5 different reservoirs of H2 in this location. And this hydrogen is used at the local scale. Basically, it is used to run an engine that produces electricity for a village. So just to finish the part of my talk, I just want to introduce briefly a work that we done in collaboration with the microbiology department of the University of Brasilia on this example. So basically here, we went on the field and we collected soil samples. And the idea was to say, okay, we have soil samples, and what about the microbiology in those soils? And the first thing that we did was to take this soil sample, put them in an autoclave together with hydrogen gas. So this is the schematic view of the setup. You have the soil sample, a couple of 100 grams. So this is closed in the autoclave. And we have a gas atmosphere in contact with the soil with 5,000 ppm of H2. And what we do is, from time to time, we collect the gas, which is a mixture of argon and H2, and we analyze the H2 concentration using a gas chromatography and see what happens. The result of these experiments is the following. Here you can see the evolution of the H2 concentration in a gas with time. So what you can clearly see on this graph is that the hydrogen concentration decreases. And okay, what is the reason for that? We think that this is related to the microbiology of the soil. And the reason for that is that when we take the soil, heat the soil up high in this locality. And what we showed on some samples that was quite interesting with our colleagues from the microbiology department is to look at the evolution of the different phyla over time. And basically, we could see a big change in the different microbiological families depending on whether the sample was fresh or if it has been incubated with H2 for a long time. So there's a kind of adaptation of the microorganism with this high H2 environment that we reproduced in our autoclaves. So just to conclude for my part of the talk, basically what I've tried to show you is that H2 can be produced abiotically through water-rock interaction. So I showed you this example in the mid-Atlantic. I show you that also in the middle of the continents, you can have H2 production, probably abiotical, but this is still unclear. But at least in the middle of the ocean, we're sure due to the temperature that is abiotical. I must confess that the global volume of this type of natural hydrogen still has to be evaluated. We don't know if there's a real resource of hydrogen that could be used for industrial application. What I've showed with the last experiments very briefly is that in those environments that are rich in hydrogen, the microbiology will be a major player in the H2 cycles. I mean, we did some calculation in the Minas Gerais structure that I showed you. Basically, if we have a source of hydrogen at depths with a concentration of 1,000 ppm, just the flux of hydrogen through 1 meter of soil column 40% of this hydrogen is going to be consumed by microorganisms. So it's huge consumption by microorganisms. And the last point I want to make is that I've showed you that the phyla content of the soils changes depending on the environment and on the presence of hydrogen. And this could possibly be used as the marker of H2-rich environments. You look at the microbiology of the soil and you can tell that this soil was exposed to hydrogen. So the microbiology could be a kind of exploration tool. So thank you for your attention, and I give the floor to Marianne.
It's okay. Do you hear me?
Oui. Yes, good.
So I'm microbiologist, microbial ecologist from the Mediterranean Institute of Oceanography at Marseille in France. And today I'm here to talk about the microbiology of natural hydrogen environments. To introduce me, my research focuses on anaerobic microorganisms involved in the sulfur cycle and also on the carbon cycle, with especially on methanogen and on hydrogen cycle, with both hydrogen producing bacteria and hydrogen consuming bacteria, such as sulfate reducing bacteria, in diverse environments, such as extreme environments, hypersaline lakes, hot springs, anoxic sediments in soil, contaminated waters, and also submarine hydrothermal vents. Today, my presentation will last 25-30 minutes, and the order of the presentation is as follows. Firstly, I will present the different processes of microbial hydrogen consumption and production in subsurface. Then I will move on research on microbial ecosystem associated with serpentinization, which produce hydrogen, and more specifically on studies on Atlantis Massif, from the Lost City hydrothermal field to the expedition IODP 357. And finally, I will finish by the Prony hydrothermal field and the associated hydrogen metabolizing microorganisms. As you can see, there are several potential mechanisms of hydrogen consumption in subsurface. In oxic condition, we can there is a aerobic hydrogen oxidation, and in oxic condition, there is many other anaerobic reaction, such as methanogenesis, sulfate reduction, nitrate reduction, iron reduction, depending on the acceptor of electron. And in fact, this different reaction can take place in different conditions, depending on the oxygen concentration and redox potential content and degrees. In fact, anaerobic reaction such as sulfate reduction using hydrogen or methanogenesis, can be performed at a redox potential condition comprised between minus 300 and minus 500 millivolts. So the different reaction of hydrogen oxidation depend of many different environmental conditions. There are also different mechanisms of hydrogen production in subsurface in absence of sunlight, such as nitrogen fixation, anaerobic carbon monoxide oxidation, and the dark fermentation, which is a step of anaerobic digestion. The last step of the anaerobic digestion, donc, here is the production of methane, and in this this process, hydrogen is a key metabolic intermediate, which can be it's a byproduct of different reaction, acidogenesis here in green, or acetogenesis. So in surface and subsurface environment, hydrogen is produced by different reaction, but often immediately reconsumed. In fact, there is many microorganisms that produce hydrogen by dark fermentation. There is strict anaerobe, such as bacteria belonging to the genus Clostridium, or facultative anaerobes, such as bacteria belonging to the Enterobacter genus. The bacteria, such as Clostridium, are mesophile, but there are also thermophile, hydrogen-producing bacteria, such as Permotoga. And hydrogenases are the key enzymes that catalyze both hydrogen production and hydrogen consumption. This is a reversible enzyme. And there are two main kind of hydrogenases. There is iron-iron hydrogenases that more specifically produce hydrogen, and there are also nickel-iron hydrogenases that are more specifically involved in hydrogen consumption. Moreover, the catalytic activities of the iron-iron hydrogenases are 10 to 100 times higher that nickel-iron hydrogenases, and 1,000 times higher that nitrogenases, and another type of enzyme that produce hydrogen. So now we will see this microbial reaction, hydrogen consumption and hydrogen production, in microbial ecosystem associated with serpentinization. So serpentinization is a natural process. It's a abiotic reaction where olivine is transformed to serpentine and hydrogen, and this hydrogen can react with carbon dioxide to produce methane, small hydrocarbons, and also a small organic acid, such as formate. And this compound, this abiotic formed compound, can be used as electron donors and carbon sources for microorganisms living in deep environments in the absence of sunlight. This reaction and this type of environment is very interesting for microbiologists for fundamental questions such as origin and limits of life on Earth and other planets, but also for applied questions such as the biotechnologic application of extremophile, because in this environment we can find alkalophile microorganisms, and it is also interesting for carbon dioxide storage and natural production of hydrogen. It is the subject of this presentation. Now I show you two examples of the research on this type of environment. In fact, we have different hydrothermal systems associated with this reaction. There is the system are present in different places around the world, and especially around the mid-Atlantic ridge, where we can find the famous Lost City site. It is a site that was discovered in the 2000s at a depth of around 800 meters below seawater. And the particularity of this site is the carbonate kinase releasing alkaline fluids, which is different from black smokers releasing acid fluids. This carbonate kinase harbor diverse microbial community, such as hydrogen metabolizing microorganisms. And the discovery of this site changed our view of the condition under which life could have emerged on our planet. This type of hyper-alkaline hydrothermal system is believed to have conditions similar to those prevail on the early Earth. However, this type of hydrothermalisms had already been reported on other places around the world, such as on continental, not in submarine environment. In continental site, we can see the Samayophyolite in Oman, or the coastal hydrothermal site in New Caledonia. We will see later the case of the Prony Bay in New Caledonia. During fall 2015, I joined an international team of scientists to take part to an expedition organized by the International Ocean Discovery Program. This IODP expedition about the James Cook During this expedition, we drill 17 holes at 9 sites using different seabed drill. So you can see the 2 types of seabed drill. And we drill this different sites across the Atlantis Massif, which is dominated by the Lost City hydrothermal field, and that produce hydrogen. And the main objectives of this expedition is to study the subsurface of the Atlantis Massif by coupling biological, chemical, and tectonical aspects. From the objective on our team of microbiologists at Marseille was to study the cultivable diversity and physiology of piezophilic and anaerobic microorganisms growing in deep ecosystem around 1,000 meters below sea level and driven by serpentinization. Our results showed that hydrostatic pressure helped to cultivate original microorganisms. We isolate a new species of the phylum Firmicutes, and this bacteria was the first anaerobic bacteria isolated from rocky subseafloor. And now I show you the case of the Prony hydrothermal field. This hydrothermal field is located in the South Pacific, in the island New Caledonia, more specifically on the south of New Caledonia. And the hydrothermal system of the Prony Bay is similar with the Lost City hydrothermal field, because active chimneys composed of calcite and brucite can be observed into the lagoon, and these chimneys discharge anoxic and alkaline fluids rich in hydrogen, methane, and other hydrocarbon. But this site is different from Lost City because it is found at lower depth than Lost City, at 50 meters below sea level, and the fluid have temperature at low temperature, and the fluid are low salt compared to the salty fluid of the Lost City. Alors, in 2011, the campaign Hydroprony, the objective of this campaign was to collect different samples in the Bay of Prony. And we collect samples from intertidal area. Here you can see an intertidal site with a chimneys, and you can see the bubble of this site. And we also collect a sample of chimneys and fluid from different sites, different submarine sites, such as the famous Needle of Prony. The analysis of the geochemists showed that all sites have fluid, have alkaline fluid with a pH of 10 or 11. And the gas composition of this different sites show that there are hydrogen that reach 40% or methane. You can see concentration around 10 or 15. And the hydrogen-methane ratio of this site is around 2. It's an intermediate ratio, lower than submarine sites such as Lost City, but higher than methane-dominated ophiolite, such as the site found in Turkey or in Italy. Alors, the objective of our team of microbiologists at Marseille was to describe the microbial diversity of the chimneys and the fluid discharged by the Prony hydrothermal field. We want to identify, isolate, and study model microorganisms and microorganisms with biotechnological interests. The second objective was to determine the role of different microbial groups in the different biochemical cycles, such as carbon cycle or hydrogen cycle, and to investigate the microbial group and processes involved in the formation of the chimneys, and the colonization of these chimneys to understand the emergence of life on Earth. Alors, for this, we use molecular and culture approaches from fluids and subsample of chimneys. We extract the DNA, and then we sequence using Illumina MySeq metabarcoding the DNA to identify the microorganisms, and we quantify the DNA using both quantitative PCR and using microscopy. We also isolate and enrich and isolate microorganisms using alkaline and anaerobic media, both in tube and bioreactors, and we monitor both the growth of microorganisms and the metabolites in liquid phase and in gas to follow the consumption and production of hydrogen and methane. Alors, this is the first result in the chimneys. We found many microorganisms and a low diversity of archaea dominated by uncultivated methanosarcinales. It's a type of archaea similar to those detected in Lost City or in a continental site. And this type of microorganisms can oxidize or producing methane, or they can also oxidize hydrogen to produce methane. We also isolate a new species of methanogenic archaea adapted to high pH and close to the genus Methanobacterium, also identified in other ophiolites. So this confirms that subsurface lithotrophic micro ecosystems can be based on hydrogen energy. Using both microbiology, microscopy, and sequencing analysis, we identify bacteria of the phylum Firmicutes as the first colonizer of the chimneys before the hierarchy. This type of Firmicutes in red on the picture could be involved in the formation and or the consolidation of the carbonate structure using organomineralization processes. And this type of microorganisms could play a central role in the ecology of this hydrothermal site. We also isolate several species of bacteria belonging to the phylum Firmicutes. They belong to different genus, and all are adapted to alkaline pH. All ferment sugars, organic acids, and amino acids. And this data will confirm by the analysis of the complete genomes. And we also isolate a bacteria able to produce hydrogen by dark fermentation. This production of hydrogen is possible at a high pH, up to 10, up to pH 10, and the hydrogen yield is comparable to the hydrogen yield we obtained using neutrophilic bacteria. So it is a very interesting finding. Using a molecular method, we analyze both hydrogen consumption and hydrogen production, and we found that proteobacteria dominate both chimneys and fluid, and that hydrogen oxidizer belonging to Hydrogenophaga genus dominate in oxic condition, and sulfate-reducing bacteria dominate in anoxic condition. So these two groups may have a role in hydrogen oxidation in the site, and using metagenomic analysis, we also found that Firmicutes could have a role in hydrogen production because they contain the majority of iron hydrogenases that could be involved in hydrogen production in the site. Finally, it's the end of my presentation, and to sum up what I said, in this side, we see we detected the presence and activity of hydrogen oxidizing microorganisms here in chimneys and in fluid. We detect a potential role of Firmicutes in biological hydrogen production, and we detect also a change in microbial community structure depending on environmental factor, including the change of concentration of hydrogen. And And the perspective is to analyze the microbial community in subsurface. The question are how does hydrogen shape the community composition and functioning in subsurface? What are the main electron acceptors used to oxidize hydrogen? What are the main microbial groups? What is the importance of firmicutes in the hydrogen concentration mixture at the surface? And the projects are now microprony, and the future proposal submitted to the International Ocean Discovery Program could be help to answer these questions. So thank you for your attention.
Thank you, Marianne.
Thanks, Marianne.
Very interesting, and Fabrice too, very interesting. So Krieger, do you want to make a comment and a question by your own before the public ones?
Yeah, actually, I will start since I have the microphone to start the discussion. I have other questions. It will be a very quick one for Fabrice and one for Marianne. And then we can start from other questions from the students, if that's okay. So Fabrice, I will make a very general question for you that's related to several ones that you are having on the chat. And can you talk to us a little bit about what is driving the companies to explore this source of energy? Since you point out several problems or several characteristics of hydrogen itself, that's difficult to compress, it's difficult to storage this gas, and it's not easy to store, right? Some people are asking if it's explosives and it can explode, etc. So what's driving the company, since it's not exactly dependent on the process, it's not exactly a green energy source. So what's driving the companies today? Why is it taking so long to develop any technology that will overcome all these possible problems?
What did you say about the development of technologies? I couldn't hear you very well.
Yes, to be able to explore commercially the hydrogen. So it's not easy to compress, and it's not easy to explore in different ways. It's not exactly green process, depending on the process. So you have production of methane and so on. So what
are the challenges to overcome problems for H2 energy in industry?
Okay, so
what are the challenges at all?
But there's already a real hydrogen economy. It already exists. I mean, there's 60 million tons of H2 that are produced yearly worldwide. So that means the technology they are there, and they already exist. And the question is, what about natural hydrogen? And that's a different thing. I mean, as I showed you, most of the hydrogen is produced through fossil fuel reforming, and this technology is mature and it works. Now, the question that some industries are interested in, and that's basically the oil and gas industries, because these industries, they are used to deal with natural gases. And as you know, they want to shift towards a new type of energy. And the idea is if you know how to handle methane, for example, it shouldn't be a problem to handle H2 because they are quite similar gases. So these companies, they are trying to shift towards, the exploration of H2, but it's still the beginning. I mean, and that's the reason why research laboratory are still very much involved, because it's it's still at the border between applied and basic research. But yes, but there are real challenges now in the exploration of H2 because it's just beginning. So now companies are investing, and you see all I've showed, these are measurements, mostly measurements that are made at the surface in the soil. But you know, if you look to gas reservoir, you should be able to drill holes and try to find the reservoir, and this needs investments because this costs a lot of money, and now the industry is starting to invest into drilling holes and trying to see what's in on the ground, basically. And this is the beginning now, and especially, I mean, in Australia, it's incredible. I mean, the industry is really much involved in trying to find a natural H2 reservoir, and they are you know, they are starting to drilling holes, and it's really beginning. I don't know if it answers your question. I couldn't.
Yeah.
Sorry.
Are you guys be able to hear me? Okay, now.
Yeah, it's a bit abafado in Portuguese.
Okay, I'll try to speak very loud. Yes. Marianne, I have a question for you, very quick, if I may. Can you already use or see a possibility to use the Firmicutes phyla as a marker, or the Clostridium genus as a marker for an environment that it would be, you know, suspect to produce hydrogen? I'm asking because Firmicutes is a phyla that is readily enriched to several different environments, especially the ones that are enriched for high levels of carbon, like animal waste, human waste, and so on. So, and in soil, of course, right? So, would it be, I already able or see the possibility to use this phyla as a possible marker for H2 production in natural environments?
I think we can't use a phylum as a biomarker. Is too large. I think we have to search more specific biomarkers, such as maybe a specific genus. We found hydrogenophaga species, but this type of species are also found in other type of environment, not only in hydrogen-rich environments. We have to continue the research, maybe by combining different techniques such as metagenomic, metatranscriptomics.
Okay. Because the question is, they are, you know, in some environments that you are studying, it seems that you have a natural enrichment for consumers. So it's a natural enrichment process, and not exactly enrichment for, or hydrogen could be possibly being produced by any of those microbial consortia. So the question is the balance between the production and the consumption of hydrogen. So it's not very easy to measure that. So I was about to ask you about metatranscriptomics, if you want, especially in marine vents.
So we got some questions from the public. I can make one and then prepare the next one.
Yeah.
I think the first one is to Fabrice. In order to increase the H2 production, it will be needed mechanic mechanisms to produce it abiotically through water-rock interaction, or green energy would be sufficient for this. What do you think?
What do you mean by increases the
H2 production? Okay.
Yes.
If you want to increase the H2 production,
okay,
it would be necessary to do mechanical mechanisms in the rocks to produce it abiotically, or green energy will be sufficient? That's the question.
Oh, okay. Well, this is a very interesting question. I don't know how to increase the natural hydrogen productions. I mean, this is geoengineering, the way to increase this. So this exists, for example, I mean, this subject is related, for example, to CO2 sequestration. So people are using rocks to try to sequester CO2. So basically what you can do is you can inject CO2 gas into a rock, and then the rock reacts and forms carbonate, and then the CO2 is stabilized and it will never go back to the atmosphere. People have been thinking of doing same kind of thing with hot water that you could inject in a rock in order to enhance the H2 production. But I think it's a little too early to think about such advanced technique when we don't really understand the way natural hydrogen is produced. So that's what people do. I mean, we did this actually. You can reproduce this natural reaction in the laboratory. So it means you can take the rock and you can expose the rock to water, to hot water, and then you produce hydrogen. So this works. But the problem is, it's an economical problem. The cost of hydrogen shouldn't be too high. And so presently, basically, it's a few dollars a kilogram of hydrogen, which is not much because 1 kilogram of hydrogen is a huge volume.
Yes.
So if you want to produce hydrogen, in the laboratory, you need pressure, temperature, water, and this costs a lot. And there's no business model that would work by doing this in the lab, because I mean, the idea, if you want to use hydrogen as a fuel, it shouldn't be too expensive. It should compare with gasoline, basically, and then you should not do, involve too much technical things that would cost a lot. It has to remain cheap.
Okay.
So Krieger, can you do?
Marianne, we have a question from a student, and he wants to know about the process of, is the microorganisms colonizing the hydrothermal vents, or they are in surrounding water? How this process is being accomplished by this community? They are actually part of this hydrothermal vent.
They form biofilms, for example.
Yes, it is the process to form biofilm, but it depends of the type of hydrothermal vents. For lost city, for example, we think that first bacteria, first archaea, or other bacteria colonize the chimneys and form a biofilm.
But this process is not economically viable, right? So the hydrogen being produced by these hydrothermal vents are not economically viable, right?
Oh, we don't know if hydrogen is the first source used by the microorganisms. Another hypothesis is that hydrogen and carbon forms abiotic organic compounds, and maybe heterotrophic microorganisms can
colonize first, but can survive first.
Maybe we don't know.
But can you imagine an experiment on that? No, just changing the conditions and then see what happens if you put someone. Then can you imagine something like that?
Yes. In bioreactor, yes, it's possible.
It's possible, yeah.
And Marianne, you said in our perspectives about fish, using fish. Obviously, you're going to use a specific probe for different groups. Why is that? You are searching for specific groups that you are already, have clues about the role on hydrogen production. You said I went with sequence to do metagenomics, metatranscriptomics, but then you said about fluorescent
to do
in vivo hybridization. So I'm interested only in those groups that are already thinking about the role and the process of hydrogen production.
Before using fish, we have to use a metavar coding to identify microorganisms in the wall of kidneys, and after we use microscopy techniques. But for now, we found many heterotrophic microorganisms. So it's surprising. At Prony hydrothermal bay, but it's a current project, and we continue the research. We collect the new sample to do this experimentation.
But is that your goal at the end is also to find out markers, biological markers for the process, or just to understand the process as a whole? We're trying to find specific organisms that can use as a marker for detecting this process happening in natural environments.
And there are several groups. We can use functional markers such as functional genes to detect sulfate-reducing bacteria. We know we know that there are hydrogenotrophic microorganisms, but it's no species and no well-known groups, and there are many uncultivated microorganisms, and we don't know the function of all these microorganisms, and it's the uncultivated microorganisms that play a crucial role in the hydrogen consumption or hydrogen production, and we cannot we don't know now the specific group involved in this type of reaction.
Okay. Fabrice, a quick question: is iron a marker or any environment samples?
Is iron a marker of any environment?
Yeah, a biotic marker. We're talking a lot about the biotic marker, right? The microbiology and the microbes as a marker. Is iron content?
Sorry, I cannot hear you. I have a problem with my. Can you? Sorry.
Tá abafado, Krieg. Tem que puxar de novo.
Maybe Andrea, you can tell me what was the question?
He's asking about iron be a marker of an environment with some kind of dynamics, microbial dynamics in generation of H2 or something like that. That's it?
Yeah.
Yeah.
Yeah. I don't know. What I can say is that we use iron as the marker of redox conditions in natural environments, and I mean that's the only thing I can say. I mean, if you if you look at the red soils that you have in Brazil, basically iron is 3+, and that's a marker.
That's why we're going to Minas Gerais, probably.
Exactly. So
Minas is rich in iron, I think.
So yes, so it's a marker of strong alteration and you know, heavy rain and things like this, and but it's a marker of redox conditions. So I don't know. Maybe Marianne has some idea about, because the iron, it could be either 2+ or 3+. And,
yes, and depends on that you have different
yeah. So under
biological reactions, we know that.
And I think in some reducing conditions due to microbiology, iron could be 2+, and you have like gray rocks related to this. So it's a marker. I mean, it's basically what Marianne showed on the different stability of metals as a function of EH. I mean, basically, iron 2+, iron 3+ is a redox marker, and this could be related to different things. Among them, the biological activity. I don't know what you think, Marianne, about this.
Yes, but I think that iron cannot be a specific marker of hydrogen. No, it's
and especially
hydrogen consumption. It's not specific. Iron is a major element,
and especially because under subsurface conditions, hydrogen is not that reactive. In the sense, if you, for example, if you have iron 3+ in contact with hydrogen, there's little chance that you will transform it in, or you will reduce it, because hydrogen, unless you have a bacterial or microorganism activity, but just, you know, H2+ iron 3+ does not make iron 2+, because the reactivity is not that high.
Can you hear me now?
Yes,
better.
Sorry about that. I don't know what's going on. So, pertaining on the same question, then, Fabrice and Marianne, one student is asking if the consumption of hydrogen by microorganisms in natural sources will be an economical problem. Now, you said that is not, very reactive, but if you have, and you know that for several different environments, especially soils, that you have enrichment for consumers, would be that a problem for the industry?
And the students are asking lots of questions concerning the possible economic use of hydrogen.
Yeah.
They are very, we got at least 5 questions in that direction.
Yeah, but I think this is really a very interesting question, because I don't know what you think, Marianne, but in most cases, when you have a hydrogen production, usually this hydrogen is consumed. You were saying, you gave some example of this, where you have producers, but oftenly when you have producers, you have consumers, and the hydrogen is consumed. Is that correct?
Yes, particularly hydrogen producing soil.
Yes.
Particularly, but because for the hydrogen produced by normal vents, maybe the concentration of hydrogen very high, and the role of microorganisms is low, maybe, but for soil the consumption of microorganisms is very important.
Yes, and for example, I know that close to leguminous roots, where you have nitrogenase, you can have locally concentration up to a few thousand ppms, but a few centimeters away, the concentration drops, because with these producers you have consumers that are associated.
Yes.
And then for the industry, this is very interesting when we go on the field with the people from the industry, and when they realized the effect of microbiology, they were really excited, because it means that if you measure like 100 ppm, then you say, okay, this is 100 ppm, but most of the hydrogen has been consumed by bacteria. So it means at depth, there is more than that. So then they are really excited. But this might be a problem for exploration. So it means, I mean, I didn't mention that, but I mentioned this example in Africa, where I told you that there is a well-established H2 reservoir in that place. So it's, and I don't know what is the depth of this reservoir, but it's not very deep. But if you measure the hydrogen at the surface, there is nothing. So I don't know what it means, but it could be consumed. But this means that for the exploration, it could be a problem, which is that even at the surface, you don't measure anything. Although hydrogen, you know, has a very high diffusivity. So normally, hydrogen,
I was thinking about it. Doesn't, exactly. You cannot, you just lost it in the.
Exactly. I mean, it should be a huge leakage. I mean, when we do experiments with H2 in a steel autoclaves, even the hydrogen can leave the autoclave, you know, because it has such a high diffusivity. So if you have some kind of concentration of hydrogen, you expect this hydrogen to be leaking everywhere. And but maybe the effect of the microorganism is to consume part of this hydrogen, and maybe you will overlook you know, an accumulation of hydrogen that would be deeper because of the effect of bacteria. So it's very, I think it is very important for exploration, this, to be aware of the role of bacteria or microorganisms.
I was thinking about your labs, both of you. You have an autoclave to cultivate microorganisms and then keep the pressure in the atmosphere, controlled atmosphere. Is a kind of very different laboratory. You have to keep the pressure, but you cannot compress so much because it explodes. Am I right? Yes.
Yeah.
So you have to keep a high temperature, but not so high. So it's very interesting. I was nice lab to visit, quite different.
So we have one last question.
Yes.
To conclude. I think that for both of you guys,
yes.
It's pertaining to that very regular pattern of hydrogen emission on the San Francisco Basin. Right? Every noon, you have that peak that happens every single day at noon. Is that a abiotic process that is happening, or a biotic process that happens every, as a clock?
Make your bets. Make your bets.
Okay.
Arguments.
You maybe I answer this question, Marianne. You don't know about these things in San Francisco, the curve that I showed. So where you have some, regular emissions of H2, which are located at noon every day, basically. So, so there was a recent paper that was published on this phenomenon, and what has been proposed is that this is due to changes in atmospheric pressure, and it's a kind of, you decrease the pressure when the temperature is high. It's like sucking the gas that you have in the soil. It's like a pump. And basically you have changes in pressure, and you can suck the gas that is in the soil pores, and that's the reason why you have these measurements. So this is one possibility that is related to changes in atmospheric pressure, a kind of pumping. And the paper I read looks quite reasonable hypothesis.
Maybe you'll be able to see that by transcriptomics.
By what?
Transcriptomics.
Transcriptomics, yes.
Okay. Yeah.
Yes.
I don't think we'll be able to see that by DNA changes during 24 hours, but maybe
Okay.
Right. For the consumer,
I cannot think San Francisco as a, you know, a hot city to do this. It would be like Sahara Desert, or even here at Caatinga, at the northeast of Brazil. I can see that because really we got very different temperatures in the night and then in the day. At noon, for example, it's quite hot to imagine that. But San Francisco is not.
Oh, well.
San Francisco based on Minas Gerais.
Minas Gerais. Okay, okay. I was thinking about San Francisco at California.
No, because
okay, that's nice. That's nice. I believe that. I believe that.
I was on the field and it was very hot.
Okay, very hot, isn't it, Fabrice? Yes, yes. That place I believe in that explanation. So, thank you very much. Do you have a last question or?
No, I think that's it. That's
yes. So
we know that's very late for you guys.
Yeah.
Thank you
very much for your time here, and hope, Marianne, you can join Fabrice at Minas Gerais and having lots of caipirinha. And we wrote at the chat the name of those ones.
Yes, yes, I wrote it on my.
Just notes. Take notes. Bring this to BH and then try all of them.
Okay.
Not at all at the same time, because we will not, you know, walk straight ahead after that. But it's nice. Okay, so thank you very much.
Thank you.
Afternoon for us to having these ideas. For me, I am an immunologist, so it's very nice to go out of the box just to think about it.
Good, excellent.
So thank you very much, guys.
See you then. See you soon. Bye-bye.
Bye.
See you.
See you. Bye. Bye.